The protein enzyme that transfers the phosphate is a protein kinase. If you're unfamiliar with this, become familiar with it. It's pretty crucial. Hyperbole alert, it's one of the most important enzymes in the cell. So it transfers phosphates often from ATP, which is a good reservoir of phosphate, onto a protein, and then a process called phosphorylation. Now, to remove it is dephosphorylation. Okay? So there's a different enzyme that does that. It's a phosphatase. So a protein phosphatase removes a phosphate from a protein, so it's dephosphorylation. It's a mouthful. And so it acts as sort of an on and off switch, depending on what you want. So it isn't always that when you add the phosphate, it's turned on. Sometimes when you add the phosphate, that means it becomes inactive, so I wouldn't rely on that. But nevertheless, it's when you add the phosphate, it's a kinase. When you remove it, it's a phosphatase. And that way, cells are capable of receiving messages from the outside. And I'm not just talking about human cells, eukaryotic cells, I'm talking about bacteria as well. And so this is remarkable. This signaling transduction pathway, if it's found and it's conserved within all of these phyla, and I might add these kingdoms and domains, it's strong, compelling evidence from a common ancestry that all life on the Earth has evolved. And so it's really, really cool. And so again, so believe it or not, this diagram is a simplification of the signal transduction pathway. But it's basically like dominoes, something like that. So if the molecule is altered, which then takes an inactive kinase and makes it active. And then the kinase, as you recall this from a moment ago, is an enzyme that is capable of transferring a phosphate. So it grabs a phosphate from adenosine triphosphate, sticks it on the protein kinase 2, which then makes that activated because it has a phosphate, so it changes its shape. And then that, in turn, activates another kinase. And in so doing, it's increasing the numbers of these secondary messages, and therefore ultimately resulting in a cellular response. Okay, so let's get into these secondary molecules a little bit, because I mentioned these kinases, but there's some other small molecules and ions that can act as secondary messengers as well. So this is pretty cool. So the extracellular, that means on the outside, is some kind of ligand. Now, it could be a growth factor, it could even be a neurotransmitter, it's something, it could be a hormone, and that binds to the receptor on the outside of the cell, and that's usually called the first messenger. Because, again, I don't want to get to be like, well, here's a discussion of secondary messengers, and it's like, well, what was the first message? The first message was the signal molecule. And then when you're in the cell, okay, so now we're in the cell, the secondary messengers can be, in addition to the kinases and phosphorolases, they could be these small, non-protein kinds of things like ions. They could be like calcium, for example, or cyclic AMP, and we'll look at that. So what they do is they participate, the secondary messages participate in the pathways that are initiated by the G protein coupled receptors, or the receptor tyrosine kinase receptors. So these are the guys that are getting it done. Okay, so cyclic AMP and calcium. And so what they do is they trigger the formation of the secondary. So these things are not normally present in high concentration. So at rest, let's just say this, at rest, the concentration of this is low. And also I might say the concentration, getting ahead of myself, of calcium is low. And so what happens is the cyclic AMP increases. And then what that does is that these kinases can receive either cyclic AMP or they can receive phosphates and they can become activated. So usually cyclic AMP activates kinases, and then that sort of transfers phosphates and gets the proteins into it. Okay? Now, obviously there needs to be regulation, so there are other further metabolic pathways that inhibit enzymes involved in these pathways, like adenylate cyclase can be inhibited by other proteins, so it goes without saying. So, an example of what I'm talking about as cyclic AMP as a secondary messenger is the G protein coupled receptor is altered, the not shown guanine diphosphate is removed, GTP is added, the alpha unit in particular is able to move over and activate the adenylate cyclase. And so, this becomes active right here, and now it's an active enzyme. And what does it do? It converts ATP into cyclic AMP, which is the secondary message. This then binds with protein kinase A, and then this thing, not shown in detail, this thing starts another signal transduction pathway, and the cell responds. So cyclic AMP is a secondary message. Now, let's talk about calcium. It's pretty important, and it's actually one of the most common secondary messengers in cells, as it turns out. It's an important one because the reason that calcium is really cool is that the cell has really good control of it. In other words, the cell tries really hard. I'll just try to make this as simple as possible. The cell can really cause calcium concentration in the cytosol to be low. You're like, well, how does it do that? Well, it spends energy doing that. It takes proteins and it pumps out calcium. And then it stores it using energy in the endoplasmic reticulum. And it also stores it in the mitochondrial matrix. And so calcium ions, along with inositol triphosphate, that's a mouthful too, inositol triphosphate, or IP3, these things act as secondary messages. And so let's get into this. So as I was describing, the cell can control calcium, and it uses energy to always pump it out of the cytosol, and stores it in the endoplasmic reticulum, or it just simply releases it. The outside of the cell is really highly concentrated in calcium. It's like 10,000 times more concentrated on the outside versus the inside. So the cell concentration is really low. So when the cell wants to do something, okay, so it wants to respond, it can then use calcium, sort of like phosphates are used. I mentioned this before, phosphates negatively charged. When that sticks onto a protein, that causes the protein to change shape. Well, calcium can do the same thing too, right? Calcium is positively charged. When calcium sticks onto something, it can cause a shape change as well. So calcium is kind of useful this way. And let me give you an example of how calcium can stick onto a protein and then cause it to change shape. So a signal is then relayed by the secondary signal transduction pathway, and it's increased by the presence of calcium in the cytosol. So calcium is going to be a really important secondary messenger because it's going to suddenly increase when the signal comes to the cell. How's that? Let's take a look. Now, on first glance, I don't know about you, sometimes I get a little intimidated, but don't be intimidated. When you look at something that's complicated or you look at the chemistry of it, don't freak out. It's like, just enjoy it. and it's a matter of time. So if you really want to know something, you can totally get into it or not. And so check this out. Let's say that this is a muscle cell. Okay, let's go there. Say it's a skeletal muscle and we want it to contract. That's the response. So how do we get a muscle cell to contract? Well, the signaling molecule might be a message from the axon terminal of a neuron, a motor neuron in particular. Maybe the secondary message, the first message is acetylcholine. Maybe it is. So if this attaches here to a G protein coupled receptor, that's going to activate the G protein, which is then going to activate another enzyme, which is going to initiate another reaction, which will produce this inositol triphosphate. And this secondary message is going to come along like this, and it's going to come like this because it's a triphosphate. And check out what it does. It attaches to a protein on the endoplasmic reticulum membrane. Now, this particular ion gate is active, meaning that it can open and it can close. And so normally it's closed. And so it can become activated or opened when IP3 attaches to it. And when it attaches to it, it's referred to as a ligand-gated active calcium ion channel. So long story, calcium comes pouring out. It's like sort of like you're opening up the floodgates, and they're coming out, and the calcium's coming out. And you're like, well, what's it going to do? What's calcium going to do? And you're like, well, it's a secondary message, and it's going to cause a cell response. You're like, well, give me an example of how calcium can cause a cell response. I will. So here's a typical muscle cell here. Now, when I say a muscle cell, I don't want to get into the detail of this, but there's many muscle cells when you think of muscle. This is the tendon connecting a muscle to the bone here. Now, these little bundles right here are called fascicles, and when you remove them like this, each one of these is a muscle cell. Muscle cell is kind of weird. It's really long, and it's tapered, and the inside of it has so much protein, so many microfibrils, that it almost doesn't even look like a cell. This yellow region right here is the endoplasmic reticulum of a muscle cell, and that's where calcium is stored. We call it sarcoplasmic reticulum when we talk about muscle. Because sarco means flesh, and everything's like sarcoplasmic, and the whole thing. It refers to sarcolemma is the membrane, and sarcomere is the sort of unit of the muscle cell that shortens. So I'm making a long story short on this, believe it or not. But how a muscle cell divides is that you have thick protein filaments called myosin, and thin filaments called actin, and those two slide past each other, and the whole muscle cell shortens. And you're like, well, how's that? Well, the myosin grabs the actin and pulls it in, and so therefore the whole thing shortens. Now, if you're following this, you're like, well, I only want a muscle to contract when I tell it to contract, and so herein lies the control. And so it will contract. It will contract when calcium is present. Now, I don't want to divert you for excuse myself in explaining this, but there's another video I produced on muscle contraction. If I can sort of kindly suggest that you watch that video, because I'm not going to go into the detail of this, but it's really cool. I'll just touch on it briefly. What prevents myosin from interacting with actin is that there's some regulatory proteins. This long thread is called tropomyosin, and this little purple guy is called troponin. What happens is the muscle can't contract if tropomyosin is blocking the active site of actin right there. It won't contract. So what calcium does is that calcium, let me draw this out. What calcium does, you know green is calcium, it's shown here in green. What calcium is going to do is interact with a protein called troponin, and it's going to change the shape of troponin, which then changes the shape of tropomyosin, which then opens the active site of actin, and then myosin is going to grab it and pull it in, and a muscle is going to contract. That's the cell response. So calcium acts as a secondary cell response messenger. And so check this out. This is sort of an illustration of what's happening here. Here's the tropomyosin, and that yellow is the protein troponin. So both those things are regulatory. And so calcium, shown in red, attaches to it, changes its shape, and therefore the muscle is able to contract. So that concludes our discussion on signal transduction pathways. I hope you enjoyed it. I hope you learned a lot from that. Thanks for watching. Hi, everyone. It's Mr. Sinti, and I'd like to talk to you today about primary growth. And primary growth is going to be a conversation that we're going to have that's going to sort of circle around the idea of apical meristems, which are, if you recall from a previous discussion, those sort of well springs of cells that are capable of mitotic division, even as the plant gets older and older and older. So primary growth is going to be about the plant growing up towards the sky and down towards the center of the earth, or up and down. But what's interesting is this primary growth is going to also lend us to a conversation about the different tissues, ground, vascular, and dermal, and it's sort of going to wrap up the conversation that we've had before about sort of differences between monocots and dicots. So all of that is going to happen in today's conversation. So without further ado, let's just attack that. And so when we're talking about primary growth, we're talking about the roots and the shoots, because this is where the apical meristems are, the terminal buds that produce up and down. And so let's just sort of jump into that and say, for example, when we look into that, let's see, let's go right into that. Let's see, sorry about that. Let's go here, and then let's go there, and there. And so here we are at the apical meristem of a root, or the terminal apex of a root. And so let me draw your attention to this region right in here. Do you see this apical meristem? If you were to go even higher under the microscope, you'll be able to see these cells undergoing mitosis. This is a good place to look. Sort of longitudinal section of a root, you can see the cells dividing. Look, if you look really closely, you can see a cell undergoing cytokinesis right there. And so right here is where the cells are dividing. But you notice here on the very tip of the root, there's something called the root cap. And the root cap are cells that are no longer dividing. Not only are they not dividing, but they're dead. And they provide sort of like a symbol, like to the root itself. Because under the soil, soil can be kind of scratchy, and it's abrasive. And so it'll tear away at the root, and those cells are sloughed off without it damaging the cells here in the meristem that are actively dividing. So the root cap plays a protective role. It doesn't divide itself, but yet it's a consequence of these cells dividing right in here. And these cells actually secrete a little bit of a slime, which helps to lubricate the root as it moves through the soil. So growth, what we're talking about is it occurs near the tip of the root. So I look down here, you see here's the root cap, and this sort of circular region right in here is the apical meristem. You see that? Apical meristem. These are the cells that are dividing right in here. There's a region in between the apical meristem and the root cap itself. This grouping of cells right in here is called the quiescent center. That's kind of a cool word. It's not restricted to biology. It just means kind of a quiet area where a reserved individual is sometimes referred to as quiescent. So it's a bunch of cells right in this area that are sort of resistant to, they're not dividing as quickly as the apical meristem, and they're sort of resistant to trouble, so they can become meristematic if called into it. And so what I want to say about this apical meristem right in here is that the apical meristem can be further divided into three kinds of primary meristem. So this is a primary meristem, meaning that it involves growth. And when the cells are dividing, the cells are going in an upward direction as they mature. And so this region right in here is the zone of cell division, right in here. So this is mitosis. And this region can be further divided into three kinds. And let's go right in here. Do you see this one that says procambion? Procambion are the cells that are dividing that will become, as they elongate, in other words, they get larger in size, and then specialize, this will become vascular tissue. So procambion matures into vascular tissue. And then once it's vascular tissue, it's no longer dividing. So notice the vascular tissue here, purple. That cylinder, which is found in the root, is called the spiel. Right in here, the spiel. Sorry for the terms, but that's the spiel. This yellow tissue right in here is ground tissue. So it's vascular, ground, dermal. Dermal's blue. Ground is yellow. So what becomes, when the cells divide, what will become the ground tissue? Well, that's ground meristem. Yay, you remember that. Ground meristem will become ground tissue. So as the cells elongate and move up, they will specialize into this ground tissue. Now, what is this ground tissue called right here between the spiel and the epidermis? This is called the cortex. If you recall, the function of the cortex in the root is that of storage. It has this leucoplast in there, it stores starch. And so that's the function of the cortex. And so the third primary meristem, right over here, shown in blue, is called the protoderm. In other words, it will become the epidermis. So as the cells divide, and then they get a little bit larger and longer in length, notice here they become the dermal tissue. The epidermis can be specialized into root hairs. Look at this, maximizing surface area for better reabsorption. So there's three zones. There's kind of they're not so distinct. The truth is there's a little overlap between the zone of elongation as the cells get longer and as they mature. There's a little overlap. So you get three zones. Let me sort of emphasize this with a pen. You get zone right here of cell division, and then the cells get a little bit larger, and then they mature, differentiate. And what are they differentiating into? Into the three tissues: the epidermis, the cortex, and the spiel. In other words, dermal, ground, and vascular. Right there. So there's three primary meristems in the root. I hope that was clear. And so here we go again, a close-up of the root. Here's the root cap. Here's the apical meristem. Here the three primary meristems are the protoderm, procambion, and the vascular cambion as well. And so those will divide through mitosis. And this region right in here is called the quiescent center. And so this quiescent center, I mentioned before, it's sort of resistant to damage. And so those cells can sort of are like bench players in a sport. They can come in, or bullpen. They can come in as the starter pitcher becomes damaged, if you will. And so these primary meristems, protoderm, Procambian and ground meristem will eventually become the three tissue types, the blue, the yellow, and the purple, right there. And so here they are, the blue, the yellow, and the purple. So protoderm, ground meristem, and procambian, right there. And so here's a cool drawing. Here's a root cap right here. Here's the apical meristem, the cells that are undergoing mitosis. Here's the quiescent center. Notice here, look at this. As the cells move upward, they're no longer dividing. So this is where they elongate. They can get really, really big. If you take a look at this under the microscope, these cells are huge. And you can see here in the center is the vascular tissue or stele, and then on either side is the cortex. If you think of it like this is a root, I don't know if you're getting the full sense of this, but if you think it's three-dimensional, this stele is a circle in the center right here. You recall that like in a carrot. There's like this central vascular tissue, and then that's the stele, and then on the outer parts is the cortex, and that's where the starch is stored. Here's the epidermis with the root hairs. Look here, here's the pits in the xylem, which will allow water moving between the cells. So this zone of maturation is when the cells begin to specialize into the different tissues, either dermal, ground, or vascular. And so what we have, again, just to sort of really drive this point home, is these primary meristems in the root give rise to the epidermis, which is right here, this outer layer of cells, this inner layer called the cortex, and then this vascular cylinder called the stele.